Bismillahirrahmanirrahim. Today we are going to start chapter number 6, Nucleic Acids and Protein Synthesis. So let's go over the learning objectives. So our objective 6.1 is we have to talk about the structure and replication of DNA. So we will be describing the structure of nucleotides, including the phosphorylated nucleotide ATP. Now the thing is, the structure formula are not required at all, right? But we will be actually talking about how they differ from each other. Then describe the structure of RNA and DNA and explain the importance of base pairing and different hydrogen bonding between the bases. And we have to include reference to adenine and guanine as purines and to cytosine and thymine as well as uracil as pyrimidine. So we'll be talking about these terms. Then structure formula for bases are not required, right? But recognition is required. Again, you can have MCQs related to that, the way you will be asked to recognize a purine or a pyrimidine. Right, so purines actually have double ring structures and pyrimidines have a single ring structure. So we should actually be looking at the diagrams there. Then we will also be describing the semi-conservative replication of DNA during interphase. So this is one stage of the cell cycle, right? Interphase, right? Where actually DNA replication takes place. So we'll be talking about all these in our objective 6.1. So let's move on to the slide then. Okay, then so let's discuss about nucleic acids and how are they important in information, how are they important as information carrying molecules basically, right? So when we talk about nucleic acids, there are two of those, right? It's DNA and RNA, right? DNA stands for deoxyribonucleic acid. And let me write the name here as well, right? So it's deoxyribonucleic acid, right? And RNA is ribonucleic acid, right? So this is how they are actually spelled, ribonucleic acid and deoxyribonucleic acid. Now, both of these are polymers. When we say polymers, it means they're made up of many monomers called nucleotides. So first we'll talk about the structure of nucleotides. Now, nucleotides themselves are made up of three main components. So if you look at this diagram here, right? So this is one nucleotide, and it is made up of three components. One of them is the phosphate group that you are seeing here. We have a pentose sugar, the one that you see in the middle. Pentose means it has 5 carbons. And then we have at one corner, we have the nitrogenous base, which can be anything from adenine, thymine, cytosine, guanine, and uracil. So these are the nitrogenous bases. So again, let me write the names here also. So A stands for adenine, right? T stands for thymine. C stands for cytosine. G stands for guanine. And U stands for uracil, right? These are the names. So it could be any of these nitrogenous bases. We call them nitrogenous bases because they contain nitrogen, right? So these three actually are like, they actually are combined together, or they actually form one nucleotide molecule as they undergo condensation reactions, because bonds are being made here and we have a bond here also, right? So they undergo condensation reactions and then one nucleotide is formed. Okay? So now if you look at this diagram, here actually they have shown you the formation of nucleotide. So here you can see this pentose sugar. I'll be numbering the number of carbon atoms in a while. Then on one side we have this phosphate group attached, right? And at one end we have this nitrogenous base attached, right? So this is actually, they have shown you three structures separately right now, because this is one nucleotide, right? And it is formed once all these three components are actually joined together, right? Or they undergo condensation reaction. Condensation reaction is the one in which water molecules will be removed, right? So here you can see, or let me first label these number of carbon atoms first. So this is how it starts. This is carbon number 1. This is carbon number 2, carbon 3, carbon 4, and this we have carbon number 5. So carbon 5 is attached to carbon 4, and here we have oxygen here, right? It's a pentose sugar. So we have oxygen here, and then this is how we start numbering it, right? Okay, so this carbon number 5 actually, right, it makes a bond with a phosphate group here, right? So what is happening? This carbon number 5 has this hydroxyl group, OH group attached as a functional group there. right? And from the phosphate, one hydrogen atom will come, and these join together to form a water molecule. Okay, so they undergo condensation reaction here, right? So one water molecule will be released, right? And they will make a bond there. Secondly, if you look here, so this is carbon number 1. So again, the OH group, that the hydroxyl group that is attached to carbon number 1, it binds to hydrogen of the nitrogenous base, right? And again, we have this water molecule removed, right? And then we will have a bond between this carbon and nitrogen now. So this is one nucleotide that you're looking at here, right? So this is one bond, right? And this is another bond here that you're looking at here. And in the same process, 2 water molecules are removed, right? So one nucleotide is formed when phosphate sugar, this phosphate, right? The pentose sugar, this one here, and the nitrogenous base. All of them undergo condensation, and then one molecule of nucleotide is made. Clear? This is the formation of a nucleotide. All right then. Now here we are talking about that how one single nucleotide then keeps on joining with other nucleotides to form a polynucleotide. Because we're talking about DNA, which is a polymer, right? DNA is made up of these nucleotides. So now let's see how these nucleotides are actually, how they join together. So what is happening, if you look at these here, so these green, the ones that you're looking at here, this is the pentose sugar, right? The one that you see here, the circular structure here, that this is actually what they have shown you as a phosphate group, right? And here, the one that you see here, the one in orange, this is the nitrogenous base. So they have actually shown you one nucleotide molecule here, right? And then you can follow, let's say if this is number 1, this is number 2, number 3, number 4. So here we are actually looking at 4 molecules of these 4 nucleotide molecules here, right? So a polynucleotide is a polymer of nucleotides, right? So as these nucleotide monomers join together, a polynucleotide will be made, right? So nucleotides join via condensation reaction. Okay? Again, the same, they will be joining to each other by condensation reaction. It means a water molecule will be removed. But in this case, now the condensation reaction is taking place between the phosphate group of one nucleotide and the sugar of the other, right? So this is how it happens. Again, let me, if I just again number it from here. Let's say this was carbon number 1. Sorry, carbon number 1 was here. But in fact, let me use a different color. So it's easy for us to identify it. Let's use a different color. So let's see if, so let me use a black one. Right, so this is carbon 1, this is carbon 2, carbon 3, this was carbon number 4, and see, this is carbon number 5. And they have marked it here also, right? I'll talk in a while why have actually they shown it as a 3'. I'll talk about that in a while. But see, so how it happens actually is, there is always, there will be this condensation reaction and a covalent bond will be formed. And the bond that is formed, we call it as a phosphodiester bond. Remember, it is being made by phosphate group of one sugar molecule, right? And the other, actually the other hydroxyl group of the other. So phosphodiester bond consists of phosphate group, okay, and it has 2 ester bonds. So this chain of sugars and phosphates is known as the sugar-phosphate backbone. So see how it is happening. So there is a carbon number 5, this carbon here, of the incoming nucleotide. So the chain is going in this direction, right? So let's say this was the first nucleotide molecule, right? So now this one, and now this is the second one. The second one would be joining to the first one here, right? So what is happening? The carbon which is attached to this carbon number 5 actually, the phosphate which is attached to carbon number 5 will go and bind with the carbon number 3 of the sugar molecule above it, right? So remember, this is 1, this is 2, this is carbon number 3, right? So the 5' phosphate of the incoming nucleotide, right? Incoming means the one which is coming, the one which is below, it attaches to the 3' hydroxyl group at the end of the growing chain, right? So let's say that the chain is going in this direction, right? If it is going in this direction, so let's see, this is carbon number 5, and this is the phosphate group attached to it, right? So this phosphate group will go and make a bond with carbon number 3, right? So this is 3'. So this is what we call as a phosphodiester bond. So phosphate, okay, and it will bond this, form this bond with OH, the hydroxyl group, which is attached to this third carbon here. So there will be OH group here. So they will actually join together. There will be a condensation reaction, right? So water molecule will be removed, right? Likewise, again, have a look here. Now, if this is carbon number 3, here we have carbon number 4, and here we have carbon number 5. See, this phosphate group is attached to carbon number 5. So see, this phosphate group now binds to, again, if we keep on numbering this, so this is carbon number 3. So this phosphate at 5' attaches to, makes a bond with this hydroxyl group of this carbon at 3'. So it will keep on happening. So it always forms in 5' to 3' direction. It always forms in this direction. 5' to 3. So at 5' we have the phosphate group, right? At 3' we have that hydroxyl of the carbon number 3, and the bond actually is made between these two. So that would be known as the phosphodiester bond. I hope it makes sense, right? Because we talk about DNA as this double helical structure, right? So as we're talking about these polynucleotide chains, so actually as these bonds are being made by this phosphodiester bonds, we call them as sugar phosphate backbone. Why sugar phosphate? Because the green one that you're looking at is the sugar here, and it has this phosphate. So phosphate and sugar, phosphate and sugar, phosphate and sugar. So we call it as a sugar phosphate backbone, right? And this is the backbone. This is the phosphate sugar backbone, right? That will be formed, right? So this is how these nucleotides, they join together to form a polynucleotide that actually makes the sugar phosphate backbone of the DNA helix. I hope it is making sense now. So remember, nucleic acids can only be synthesized. That's the rule, right? It can only be synthesized into 5' to 3' direction, right? Okay then. Actually, let me also tell you, okay, what does this mean, this 5' and 3'? The thing is, this nitrogenous base also contains carbon, right? So if these are the carbon here, then we are like numbering them as carbon number 1, 2, 3, and 4, and 5, right? So since these are also carbons, so just as a differentiating purpose, we just simply use the term as 1' for carbon number 1, 2' for carbon 2, 3' for carbon 3. So remember, as a rule, nucleic acids can only be synthesized in this direction, 5' to 3'. And I hope now you know what does it mean by 5' or 3'. So these are the numbering of the carbon atoms, right, in the pentose sugar, basically. All right then. So DNA, which is deoxyribonucleic acid, and RNA, which is ribose nucleic acid, they are nucleic acids, right? DNA mainly stores the genetic information of an organism, while RNA helps in the process of protein formation. So as we complete this discussion about the DNA replication, we'll move on to how protein is synthesized, and then the role of RNA will come. So both of these are made up of sugar, right? So sugar is the ribose sugar, right? But there is one difference between them. So since we are talking about DNA and RNA, right? So difference is there in the sugar basically, right? So in DNA, it is known as deoxyribose, right? In RNA, it is known as ribose. So in the next slide, I'll just show you what is the difference between deoxyribose and ribose sugar, right? The nitrogenous bases, right? Like in the start of the lesson, as I said, okay, we have 5 nitrogenous bases, right? So we have adenine, guanine, cytosine, thymine. But in RNA, there is a difference. Thymine is replaced by uracil, right? So at one time, there will be 4 nitrogenous bases, right? But in RNA, we do not have any thymine. There is uracil in place of thymine, and uracil is not found in DNA. So nitrogenous bases can either be purines or pyrimidines. So we'll talk about these two terms also. What does this mean by purine and pyrimidine, right? So adenine, guanine, okay, both of these are known as purines, while cytosine and thymine, as well as uracil, they are known as pyrimidines. So there's a way to remember them also. We'll talk about that. So, and one more difference between them, purines have 2 rings, while pyrimidines have 1 ring, right? So this whole nitrogenous basis structure we'll discuss as we look at the diagrams. It will become more clear to you. And both these DNA and RNA will also have a phosphate group attached, right? And phosphate group remains same for both the nucleic acids, right? So remember, DNA, RNA both have sugar, okay, which is a pentose sugar, right? But it's a deoxyribose in case of DNA, hence the name deoxyribonucleic acid, and is a ribose sugar in case of RNA. Again, RNA ribose. Nitrogenous base, adenine, guanine, cytosine will be same in both the cases, right? But thymine will be present in DNA, while uracil will be present in RNA, and phosphate group is same for both the nucleic acids. Okay. Now, this is what I meant by ribose and deoxyribose. So let me actually show you these structures then. Now, here again, if you look at these structures, here they have written as ribose, here they have written as deoxyribose. So the main difference between them is this. At carbon number 2, there's a hydroxyl group attached, right? So this what makes it a ribose sugar, right? In deoxyribose, at carbon number 2, we only have hydrogen, right? So just like remember it like this, like you have heard the term about deoxygenated blood. Deoxygenated blood is the blood that doesn't contain oxygen. See, so here we don't have any oxygen here, right? So this is the difference between deoxyribose. So this pentose sugar would be found in DNA, right? Exclusively in DNA, there will be no oxygen there, right? But when we will talk about in terms of like RNA or ribose, so ribose has this O also attached to say hydroxyl group attached. So there's one difference that you can be asked of in your MCQs to identify, right? And here they have shown you the same numbering, carbon number 1, carbon 2. So you have to look at carbon number 2, right? So look at carbon number 2, and if you find OH, so it has to be a ribose sugar. If you find only H, it's a deoxyribose. Then remember carbon number 3 is important in terms of, okay, this is the OH, which will bind to the phosphate group of the 5th carbon, okay, of the next molecule. So here you can see, so this is carbon 4 and this is carbon number 5, right? So pento, jo phosphate group hoga, it will actually be attached to carbon number 5. So you can see it here also, right? So we have to actually take into account of these carbon number 2, importance of carbon 2, okay, carbon number 3 and carbon number 5, right? Okay then, so we just talked about a while back that bases are divided into 2 main categories, purines and pyrimidines. So there is a way to just remember them also. Now purines are adenine and guanine, and how you can remember them is simply as all gods are pure. So all adenine, gods, G stands for guanine, right? So adenine and guanine, pure from purine, right? So there's another way to like remember them also. Purine ko you can also remember by means of, because we'll be looking at the structure. Sorry for that. Yeah. Yeah. So purines ko you can actually also remember through the way of like purine has this U, right? And you can actually like just a way to remember it as double. Okay, double also has a U, because when we talk about purines, we are looking at the double structures, double ring structure. So the one that you are seeing here. Okay, so purines have a double ring structure, and we talk about pyrimidines. Pyrimidines have a single ring structure here. So we look at the diagram next to it, right? But this is another difference. So purines, just a way how you will remember that purines are adenine and guanine. So just remember it like this: all gods are pure. So pure for purine, and here A stands for adenine, G stands for guanine. Then you can decide definitely, because we have 5 of the bases: adenine, guanine, cytosine, thymine, uracil. So whatever is left, definitely that is pyrimidine, right? Okay, then one more thing. Do not confuse adenine with adenosine in ATP. Okay, so that is different. The structure is different. Adenosine, okay, don't confuse it with that. Again, like you need to write the correct spelling there also. And thymine, never ever confuse it with thiamine. Thiamine is actually a vitamin, right? So do not confuse it with thiamine. It is not thiamine, it's thymine. So make sure you're using the correct vocabulary terms there, right? So it's adenine and guanine, which are purines. They have double ring structures, and pyrimidines are the remaining of those: cytosine, thymine, and uracil, right? And they have single ring structures, right? And now here you can see the difference between these. See, so here they have shown you one nucleotide. Now remember, it will have this sugar, right? Phosphate group will be same in both DNA and RNA, and then there will be a nitrogenous base, and then we can have any of the nitrogenous bases from purines and pyrimidines. Now, if we zoom in, the sugar can be a ribose sugar or a deoxyribose sugar, right? So what are we looking at? Carbon number 2, right? Here in deoxyribose, look at carbon number 2, we have only hydrogen. And here at ribose, at carbon number 2, we have hydroxyl group. So this is what you have to look at. This is how you differentiate whether we have the sugar or the nucleotide that is given to us belongs to a DNA or RNA, right? Phosphate group remains same for both, right? So they have shown you here. Now coming to the bases. Purines, remember we said all gods are pure. So, so, adenine and guanine, look at the double structure. So we have this double ring structures here. So this is adenine and guanine, which are purines. They have double ring structures. And pyrimidines are the remaining of those: cytosine, thymine, and uracil, right? And they have single ring structures. So for purines. And when you look at pyrimidines, we have the single ring structures, right? So it's thymine, uracil, and cytosine. So just a way to actually remember these, because again, you're not supposed to draw the structures, right? But you're supposed to actually recognize them, right? So again, your exam tip says you don't need to know the structure formula of these bases, just which are purines and which are pyrimidines. This is what your examination actually, the specification says. You're not supposed to know and draw the whole structure formula of these bases. Okay, with that, let's discuss about ATP also, because sometimes like students get confused between like adenine, right, nucleotide, or ATP, right? So look at this. See, we have this ribose sugar. Remember, ATP is different, right? When we talk about a nucleotide containing adenine as a nitrogenous base in DNA. Remember, DNA mein the sugar was deoxyribose. It was not ribose at all. Remember that. That's one difference, right? So it's a ribose in ATP. Then adenine is the same nitrogenous base, right? But in this case, when we talk about ATP, we have 3 phosphate molecules attached here. Okay, so that's why we call it as adenosine triphosphate. Adenosine triphosphate. See, the adenosine is these two, the ribose sugar and the adenine. These two combined, they're known as adenosine triphosphate. It has 3 phosphate groups attached, right? And ADP, again, whenever there is some catabolic reaction takes place, okay, energy is released. Energy is released, it means one phosphate molecule is released, right? One bond is broken, so we have now left with 2 phosphate groups, so we will call it as adenosine diphosphate, right? So energy is released here. And while it's like anabolic processes, right? Because like, you know, okay, energy is actually required there, right? So these anabolic reactions, they store energy released from catabolic reactions. So in this case, this phosphate will be added to it, right? So energy will be required there, right? So energy is required in these processes, catabolic, and the energy is released. And then the difference is that in RNA, when we talk about protein synthesis, remember RNA ke andar, what will be the different? This base will be different, right? We will have uracil, right? It will not bind to thymine, right? And adenine, okay, when it binds to this part here, okay, so adenine forms adenosine when it binds to a ribose. Remember, only ribose. So this is what we see here, adenine plus a ribose. Both of these collectively will be known as adenosine. We call that a nucleoside, right? And when it becomes attached to this deoxyribose, then it becomes deoxyadenosine. Okay, तब हम इसका नाम adenosine नहीं बोलते. We call it deoxyadenosine because it is attached to a deoxyribose. ये इस केस में होगा जब ये DNA के साथ लगा होगा. ठीक है? It forms adenosine triphosphate and nucleoside triphosphate when 3 phosphate groups are attached to adenosine. So remember, अभी जब हम nucleotide का structure देखेंगे, the nucleotide only has 1 phosphate group. Remember, we just saw that in the previous diagrams also. We have only 1 phosphate group. Okay, this is your nucleotide, only 1 phosphate group. And if you're differentiating from ATP, remember ATP has 3 phosphate groups attached to it. Okay, then make sure you are looking at this structure. This is a ribose, right? So make sure you're looking for these things there. Now it has nothing to do with nucleotides, right? But it's just there in your book also. So just to make you know the difference between ATP and the nucleotide. All right, then let's talk about the structure of DNA. So this is an overview, and then we talk about this in detail also. So DNA is made up of 2 polynucleotide chains in a double helix structure. So when we talk about 2 polynucleotide chains, so remember this is 1 strand that you're looking at here, right? So this is 1 polynucleotide. This, remember we just talked about the polynucleotide which are made from nucleotides as they actually went this condensation reaction and form phosphodiester bond between them, right? So this is 1 strand or sugar phosphate backbone, and this is another one, right? So DNA is actually made from 2 polynucleotide chains in a double helix structure. Now 2 DNA strands. Now these polynucleotide chains, they actually have to be they have to join with each other also. So these strands join together by hydrogen bonding between the bases. And these bases, as I'm talking about those nitrogenous bases, adenine, guanine, cytosine, thymine, right? I'm not using the term uracil here because we're exclusively talking about DNA right now, right? So these bases, right, they will join together. And remember, purines always make a bond with pyrimidines, right? So it will always be a purine and a pyrimidine. That will actually make a hydrogen bond, right? So remember purines में, if we're forgetting the names, remember from the statement all gods are pure. So adenine and guanine are purine, right? So in the remaining we have this cytosine and thymine. These will be pyrimidines, right? So how it happens, adenine always pairs up with thymine, always, right? So thymine is a pyrimidine, this adenine is a purine, right? So adenine will always make a bond with thymine, and there will be double hydrogen bonds between them, 2 hydrogen bonds. So it's adenine thymine, it can be thymine or adenine. Likewise, guanine, which is a purine, right? It will bind with cytosine, and there will be 3 hydrogen bonds between them, right? And then it can be other way around also, cytosine or guanine. So remember, it's always a purine and a pyrimidine, right? Purine, pyrimidine. So this is how it goes, right? So each base can only join with one particular partner, and this is what we call as complementary base pairing, right? So this is one base, this is another base, and as they actually like join together by hydrogen bond, we call them complementary base pairing, right? So these are complements of each other. Now, adenine always pairs up with thymine, we discussed. Cytosine always pairs with guanine, right? It simply means there will always be an equal amount of adenine and thymine in a DNA molecule and equal amounts of cytosine and guanine. It means, like, since they like purines are binding with pyrimidines and adenine is only binding with thymine. For instance, I say if there is 20% adenine in a DNA molecule, it means thymine will also be present in the same percentage, right? So if adenine is 20%, thymine will also be the 20%, right? So that together actually, so remember we have 4 bases, right? So if 2 of them are making up 20 plus 20, 40%, so 2 bases have actually worked out 40% here. That actually then remaining is, the left is 60%. Now this 60% will then be divided between cytosine and guanine. So if someone asks you that how much will be the percentage of cytosine or guanine, so you can simply say 30% cytosine and 30% guanine. Because if you're making this 100%, right? So cytosine and guanine will be equal in like amount or proportion, while adenine and thymine will be equal. So I hope you get the point between when we say that adenine and thymine or cytosine and guanine will always be there in equal amounts, right? Okay, then we have already talked about that there will always be 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between cytosine and guanine, right? Now, this is important here. These complementary base pairing ensures that the replication of DNA is accurate, right? So this is important. It will also ensure accurate transcription of genes for protein synthesis. Protein synthesis, right? So we'll talk about protein synthesis and we'll see how this the messenger RNA is actually formed there by the process of transcription, right? So for now, for DNA, just understand complementary base pairing is very, very important, and it ensures, so make sure you're using this term there. It ensures that replication of DNA is accurate, right? We need it actually make sure that adenine only binds with thymine and nothing else. Cytosine only binds with guanine and nothing else, right? So for this accuracy, right, this complementary base pairing actually ensures this fact, right? But the thing with hydrogen bonds is that hydrogen bonds, when we talk about them, like on their own, they are very weak, right? But since they're weak, so it allows actually these DNA to be separated, because as DNA has to be replicated, right? It first separates. This double helix actually it opens up, right? So as it opens up, as it is opening up, so these bonds between these complementary bases, actually these hydrogen bonds get broken. So we'll talk about how it gets broken in detail in a little while, right? But these bonds have to be broken first, right? So, and as these bonds break, the DNA, these double-stranded DNA helix, it gets separated, right? And then replication can occur. And further, like when we talk about protein synthesis further, transcription can also occur, right? Now, remember, I just said this is a brief overview. We'll talk about all these in detail. Then when we talk about these sugar phosphate backbones, these actually always run anti-parallel to each other. Anti-parallel means if one is going in one direction, the other would be going in opposite direction, right? So we'll see how this happens, right? So these always run anti-parallel, and the polynucleotide strand twists in the opposite direction to form the DNA double helix, right? This DNA double strand is important as each strand can then act as a template in replication, a process which is known as semi-conservative process. So there is a method of DNA replication, okay, the one which is accepted now. So it's semi-conservative process. So this opening up of this double-stranded structure, okay, then we have one strand each, the one parental strand each, which acts as a template. Template का मतलब है something that you can use as a pattern, ठीक है? उसके ऊपर दूसरा नया आपका DNA का strand बनेगा, ठीक है? So it acts as a template in replication, which is known as a semi-conservative process, meaning that DNA is copied accurately. Okay, so now have a look at this diagram here. So here what they've shown you, the one that you are seeing in this light yellowish, brownish color here, right? So here they have shown you the sugar phosphate backbone. And remember, it was being formed as these nucleotides were forming a polynucleotide. We discussed that earlier, right? So how it was happening? So this phosphate group, which is attached to carbon number 5, right? It makes a bond with hydroxyl group of carbon number 3. So it makes a phosphodiester bond, right? So it's always from 5' to 3', right? So see here, 5' to 3', 5' to 3'. So this is the sugar phosphate backbone. So the structure is like that. So this is the sugar phosphate backbone, and the nitrogenous bases are always projecting outside or outward. Okay, हमेशा बाहर की तरफ project कर रहे होंगे. So this is the nitrogenous bases. So they have shown you here for adenine, guanine, thymine, and cytosine, right? So these are 4 nucleotides that I've shown you, right? And they have this 5' to 3'. So this is the direction of this formation of this polynucleotide chain, is always from 5' to 3', right? Okay then, and then you can see adenine, as we said, adenine binds with thymine through double hydrogen bonds or 2 hydrogen bonds. So you can see here, right? Then we use the term that these are complementary base pairs. So you can see, look at the structure, look at this part of adenine, and then look at this part of this thymine. So they actually are complementary to each other, right? Thymine will fit exactly to adenine. Then look at this guanine and cytosine, right? The thymine and adenine. So they actually be forming 2 hydrogen bonds, thymine and adenine, or adenine and thymine. And if you look at guanine and cytosine, we have 3 hydrogen bonds. So we have 3 hydrogen bonds there, right? So here they have simply shown you the same nitrogenous bases, because just use it as a key. So look at the structures, right? So this is the sugar phosphate backbone. Okay, one more term that I use, I said that they actually run anti-parallel to each other. So if you are looking at this sugar phosphate backbone here, and I clearly said that the nitrogenous bases have to face outside, right? Or outward, or they have to project outward. बाहर की तरफ होना चाहिए, ठीक है? बाहर की तरफ क्यों होना चाहिए? Because again, how would they actually make a pair? अगर उनको base pair और के साथ attach होना है complementary में, they have to make hydrogen bonds. We need to have another base pair here, right? So I use the term that they run anti-parallel to each other. So see, look at this part here. See here. Remember we said that the phosphate group is attached and they always run in the 5' to 3' direction. So if it is sort of like inverse हो गया, inverted direction में अगर ये चल रहा है, so it is going in 5' to 3' in this direction. ठीक है? 5' to 3' यहाँ पे चल रहा था. So look here. This is the phosphate. ठीक है? 5' phosphate, and it is binding to the 3', this carbon here. ठीक है? So 5' to 3', 5' to 3', right? But remember we said okay, this nitrogenous base will always project outside or outward. So nitrogenous base, ठीक है, is attached here. ठीक है? So see, it is projecting outside. Projecting outside. So अगर ये चीज नहीं होती, तो how would the bonds be made? ठीक है? I'll show you another diagram for that to make it make the understanding more like easy for you. Now see here, it is the same diagram जो अभी पीछे आपने देखी. ठीक है? Exactly the same. What I've done is इसको मैंने 2 halves में cut किया. ठीक है? So just to show you कि why it is running anti-parallel. See, if अगर मैं इसी same diagram को, let's say, because this is the 5' to 3' direction, it is going in this direction. अगर मैं इसका twist करती हूँ, let's say if I rotate it, ठीक है, and I move it like this. So had they been running parallel to each other, to see what is happening in this case. अब यहाँ पे भी देखो जो आपके nucleotide जो bases है, nitrogenous bases, they are facing outwards. They are projecting outwards here. ठीक है? If you see the same the sugar, the phosphate, right? This sugar phosphate backbone here, and if another one was here also, अगर मैं दोनों को side by side भी करूँ, तो how would or how was it possible for this adenine to pair up with thymine? How could it be possible, right? So to avoid this actually, so this is the reason actually that they run actually anti-parallel to each other, right? So if I actually now twist it back again, so see with it. So it is only possible these nitrogen bases or their pairing and making hydrogen bond with each other is only possible when the sugar phosphate backbones are running anti-parallel to each other. So if one is going in this direction, other would be coming in other direction. I hope it makes sense now. So we said that DNA is a polymer made out of monomers called deoxyribose nucleotides. And I hope you remember that deoxyribose means there is no oxygen attached to carbon number 2, right? And we have 2 polynucleotide chains. So now you are looking at, this is chain number 1 and this is chain number 2, right? But it actually, it makes a double helical structure here, right? So you can look at the sugar phosphate backbone, right? Which is made up of sugar and phosphate attached to 5' carbon there, right? Now each polynucleotide is made up of 4 types of nucleotides. So we discussed that. Now we only use the terms guanine, adenine, cytosine, and thymine, but in fact, the full name is actually guanine deoxyribose phosphate. So remember, it has only one phosphate group attached to it. All of these nitrogenous bases, or this nucleotide, in fact, right? It has one one phosphate group actually attached to it, right? So it's not like 3 phosphate groups as in ATP, adenosine triphosphate is only one phosphate group. Right? So similarly, adenine deoxyribose, we are calling it deoxyribose. Remember, these are part of DNA, right? Adenine deoxyribose phosphate, cytosine deoxyribose phosphate, thymine deoxyribose phosphate. So we know these nucleotides belong to DNA, right? And phosphate means there is one phosphate group, only one, right? So these nucleotides, they undergo polymerization. So we saw that. And then through this condensation reaction, there's phosphodiester bonds are being made between the nucleotides, right? Okay then. So yes, we discussed that this is a condensation reaction. So here they have shown you this phosphodiester bond here. So this O bond here. Remember, this carbon had hydroxyl group also attached, right? So the H got removed from here, and from the phosphate, it got removed, right? And we have this phosphodiester bond, right? So a water molecule will be removed. So during this, the sugar group of one nucleotide combines with the phosphate group of the other, or adjoining nucleotide, forming a phosphodiester bond. We have already discussed that earlier. So here what they've done, they've shown you the same structure, but in a horizontal way, right? Instead of a vertical chain, they've shown you the same as a horizontal structure. So what they've shown you here, this is actually, these here are actually the phosphate sugar backbone, right? So here they have shown you this 5' to 3' direction. They're again running in this direction. So you can see here, this phosphate group is attached to carbon number 5, right? And then like carbon number 3 is attaching to here. So you can see they have shown you the direction here, and they have shown you the nitrogenous bases, right? So these bases actually, remember, they are always be projecting outwards, right? So they have shown you the bases here, and they have shown you the bonds here also. So adenine would definitely be combining with thymine here, but there are 2 hydrogen bonds. Guanine would be definitely combining with cytosine. So you can see 3 hydrogen bonds there. So it's thymine, so it's adenine, thymine, cytosine, guanine, cytosine. So there will be complementary bases. Right? So these sugar and phosphate groups are arranged linearly. So you can see they're arranged in a line, right? And alternatively. Alternatively means we have phosphate sugar, right? Or you may call it a sugar phosphate, right? So they go alternatively. And nitrogenous bases will always project outward from each sugar group. Okay. So this is just a recap of what we have already discussed, right? So DNA is made up of 2 such polynucleotide chains, right? The chains are united by means of pairing nitrogenous bases. And we know that these nitrogenous bases, they join up through hydrogen bonds, right? And it will always be one purine combining with one pyrimidine. And so just remember it through this mnemonic, all gods are pure, right? So adenine and guanine are purines. So adenine will always pair up with thymine, guanine will pair up with cytosine, but remember there has to be hydrogen bonds, right? And these pairs are known as complementary base pairs, right? So we just talked about there will be 2 hydrogen bonds between adenine and thymine and 3 between guanine and cytosine, right? And these polynucleotide chains, they lie parallel to each other, but remember they lie anti-parallel. If one is going in this direction, 5' to 3', right? The other would be coming in this direction. So here it would be the 5' to 3' direction, right? Okay then. Now this whole helical structure was actually presented by 2 scientists, which are known as, the names are Watson and Crick. And then hence we call it as a Watson and Crick model. So this is how actually they set these 2 chains, they wrap around each other and they obtain a helical formation, right? And that's why this structure of this whole molecule of DNA, as it is running anti-parallel to each other, it is known as a double helix molecule. So these are these 2 scientists actually that actually their work led to this whole structure, helical structure about DNA. Okay, so let's talk about the functions of DNA molecule, right? So DNA basically stores the genetic information. So we know it has this genetic information there, right? So it stores that and then it conveys this information to the other generations, right? It controls all the metabolic reactions within the cell. And when we say metabolic reactions, because like it actually codes for proteins. And when we talk about metabolic reactions, we are talking about enzymes, which are also proteins, right? So all metabolic reactions in the body are actually controlled by enzymes, right? And it also creates new variations due to ability of changing itself, right? So the coding of this sequence of amino acids can also change, right? So it creates new variations due to ability of changing itself, right? So these are a few functions of DNA. Okay, then let's now talk about DNA replication, how DNA replicates, right? So DNA has this ability, it can replicate itself, right? And you must remember from chapter 5, as we were talking about mitotic cell division, so we talked about the cell cycle. And in the cell cycle, there is one phase which is known as interphase, right? Interphase had 3 stages. So there was a G1 phase, the growth phase, right? We had S phase. S phase was the one where actually the DNA replicates. And then we have the G2, right? Where actually like, if the DNA has replicated, and if there is any error there, right, so it will be corrected, right? And then after that, it will undergo the nuclear division, which can be mitosis or meiosis. And after that, cytokinesis will be happening, right? So this DNA replication occurs during the S phase of interphase, right? I hope you remember from the last chapter. So DNA, as it replicates, it has to copy itself before cell division. Remember, it has to do it before cell division so that each new cell has a full, complete amount of DNA, right? So this is what is actually called the semi-conservative replication, that the actual DNA, that the parent molecule actually opens up, right? It it opens up because it's double helical structure. So half of the strand would contain the new DNA molecule and other half would be the original DNA molecule because it opens up, right? So by doing this, it ensures, so make sure you'll be using this term, it ensures the genetic continuity between generations of cells. And these are the cells which are produced by cell division. They will inherit their genes from parent cells, right? So this is how it replicates. Okay then, so this DNA replication takes place in 3 stages. And remember, we are talking about 2 identical molecules. These are 2 identical molecules that are made from the mother DNA molecules. So if you're looking at this one, this is the original strand, right? And it opens up. So we were using the term semi-conservative. So semi-conservative is one strand would be from the parent or from the mother DNA, and the other would be the newly formed strand, right? So that would be the new one. So we call it as a semi-conservative. So 3 steps are involved in DNA replication. The first one is unwinding of the mother molecule, right? So this mother DNA strand will unwind. So we look at how it is actually unwinding, right? So it actually becomes 2 separate strands. Then assembling of the complementary bases. Now, as a new strand will actually form here. So remember, these nitrogenous bases, they actually have to bond with each other, right? So assembling of complementary bases, then formation of new strands. And eventually, now these sugar phosphate like backbone will also be made and new strands will be formed. So these 3 are the steps, right, which take place during DNA replication. So let's talk about them one by one. So the first one is unwinding of the mother molecule. So again, a signal comes that DNA has to replicate. So this happens, remember, during the interphase, the S phase of interphase. So this DNA double helix, it unwinds, right? It would unwind. So you can see this is the helical structure and it is unwinding from this, place here, right? So it actually unzips or unwinds, and this takes place by means of an enzyme, which is known as DNA helicase. So you can again, just easy to remember, DNA is a helical structure, right? Remember, these complementary bases were actually joined together by hydrogen bonds. So this DNA helicase actually breaks those hydrogen bonds, right? And hydrogen bonds are weaker. So DNA helicase, it unzips or unwinds the DNA by breaking those hydrogen bonds, right? And what happens because of that, these nitrogenous bases are now exposed, right? Initially what happened, they were actually binding to each other by hydrogen bonds, but since the hydrogen bonds are no more there, they will be exposed now, right? So we have one strand here and the other parental strand here, right? Each strand will now serve as a template to form new strands. And when we say template, it simply means it forms as a pattern. So all over this pattern, a new strand will be made. Okay, now one more thing that you need to remember is that there is not that this DNA helicase actually it will not split this entire molecule like in one go. ठीक है, ये थोड़े steps के अंदर होता है. ठीक है, so what happens is DNA molecule it will never ever be split into two steps separately. ठीक है, two strands इस तरह कभी भी नहीं बनते. ठीक है, so it will be a stepwise approach. So if you see here, ठीक है, so it keeps on opening, unzip. ठीक है, unzipping time के साथ साथ थोड़ा करके. ठीक है, so एक ही गो में कभी भी सारा का सारा strand नहीं खुलता. But what happens as the time as it is being opened, so at the same time यहाँ पे दूसरा strand है, it keeps on forming also, right? So this was the first step. The DNA helicase, so remember the name of the enzyme, it would unwind and unzip the DNA, and what it is doing, it is breaking the hydrogen bonds between the complementary base pairs, right? Now comes the second part, which is assembling of the complementary bases. So what is happening because of this unwinding, see these base pairs are now exposed. These are now exposed, right? So now they will be actually binding to complementary base pairs. So what happens in the nucleus, there are free nucleotides, right? So we know these are nucleotides as adenine, thymine, guanine, and cytosine. We are talking about DNA, right? So these and these nitrogenous bases will be there in the nucleus, right? They will be there. So what they've shown you here, they have shown you these bases. So the pink one that just showing you is this sugar phosphate, right? This is one nucleotide, right? So nucleotide has this this sugar, pentose sugar. It is attached to phosphate group, and then they have shown you this nitrogenous base there. So this is adenine. So you can see complementary shapes there. So this is adenine. So thymine has this complementary shape. It will bind to it. Then you can look at guanine and cytosine, so complementary shapes. So in the nucleus, they're actually freely floating, these free nucleotides, right? So what happens, like they will, as the DNA has to replicate, and which happens during the S phase of interphase, these nucleotides will become activated. Now, when we say activated, it means two phosphate groups will attach to them. Remember, there's always, there is still one phosphate group attached to them. The whole one nucleotide has one phosphate group attached to it. Besides that, two more phosphate molecules or phosphate groups will be attached to them. This is actually, this will make this nucleotide activated. Okay? As they become activated, now they will go and bind to the DNA strand, the one which is actually the one which has this exposed nitrogenous bases, right? So it will go and like make complementary bases with these. Okay then. So these free nucleotides with three phosphate groups are known as nucleoside triphosphates. So remember, it will be with all four of these. Okay, so adenine, thymine, guanine, cytosine. So as they become activated, each of them will have two phosphate groups attached. So we call them an activated nucleotide. Now, the extra phosphates activate the nucleotides, enabling them to take part in DNA replication. Okay, so they can only take part in DNA replication when they are activated. So the bases of the free nucleoside triphosphates align with their complementary bases. So for example, अब अगर adenine है, that will definitely combine with thymine. ठीक है, if it is there thymine on the exposed strand there. ठीक है, if it is guanine, so cytosine will combine with that. And then they will actually again make the same hydrogen bonds. So adenine and thymine, or thymine and adenine will make double hydrogen bonds, and triple bonds will be made between cytosine and guanine, or guanine and cytosine, right? Now, all these processes are catalyzed by DNA polymerase. Now, as these new hydrogen bonds are being made, this step is being catalyzed by DNA polymerase. So remember, when we talk about DNA helicase, it was unwinding the DNA double helix. ठीक है, वो खोल रहा था वहां पे। DNA polymerase क्या कर रहा है? It is actually like joining these complementary base pairs, ठीक है, by through hydrogen bonds. So this is the second enzyme that you need to remember, right? Now, just remember these activated or phosphorylated nucleotides are simply like ATP stands here for, this is adenine, right? This is for thymine. Remember, we have now 3 phosphates, so these are activated forms. Cytosine, right? And this is guanine. So we have 3 phosphate, triphosphate here. But what is happening? There's a structural difference between these phosphorylated nucleotides and the nucleotides in the DNA molecule. And why we are saying that? Remember, the nucleotides in the actual DNA molecule will have only one phosphate group attached to them. Remember, there's only one phosphate group attached to these nucleotides, right? And to the ones which are originally attached to the DNA helix, right? The ones which are attached here, there's only one phosphate group, right? But these activated ones, right, they have 2 extra phosphate groups. So remember this difference. Okay, and here they have shown you, this was the original parent strand, ठीक है, DNA helicase की वजह से, so this is one strand, this is another strand, right? And then what happens? These are the complementary base pairs. Now this यहाँ पे अब इनके बीच में जो bond बनेगा, it will be catalyzed by DNA polymerase, right? So this will be catalyzed by DNA polymerase, और ये activated nucleotides होंगे जो आके यहाँ पे attach होंगे, ठीक है? और जब ये attach हो जाएंगे तो जो extra phosphate group इसके साथ attached है, they will be removed, ठीक है? They will again go back, ठीक है, and they will then get attached to other nucleotides, वो उसको एक्टिवेट कर देंगे, ठीक है, और इस तरह वो दोबारा वो जो नए वाले आएंगे वो नई जगह पे आके स्ट्रैंड पे लग जाएंगे, ठीक है? So this is the process of assembling of complementary base pairs. Now, the thing is, अब it's not like that कि DNA helicase इसको जैसे ओपन किए जा रहा है तो एक टाइम पे एक ही एंजाइम काम कर रहा है, because what will happen, both of these enzymes are working. Like for example, if DNA helicase is a stepwise, it is unwinding this, ये original strand है, ठीक है, if it is unwinding it, ठीक है, इसको unzip कर रहा है, तो at the same time यहाँ पे, ठीक है, complementary base pairs भी आके बनना शुरू हो जाएंगे. So DNA polymerase will start working here, ठीक है, it will start working here, right? So this is what will happen, right? So these two enzymes, ऐसा नहीं है कि like action of these two enzymes are completely exclusive, ठीक है? DNA helicase, as it is moving along the molecule, breaking the hydrogen bonds and being closely followed by DNA polymerase, assembling the new strands. तो जैसे ओरिजिनल वाला ब्रेक होता जाता है, यहाँ पे न्यू स्ट्रैंड्स जो है, जो आपके न्यू कॉम्प्लीमेंट्री बेस पेयर्स है, वो असेंबल होते चले जाते हैं, ठीक है? सो इस तरह दे आर वर्किंग नाउ. द रिजल्ट इज देन DNA मॉलिक्यूल टेक्स अ शेप दैट इज रेफर्ड टू एज अ रेप्लिकेशन फॉर्क, ठीक है? सो इफ यू लुक एट इट, यहाँ पे तो ये स्ट्रैंड, ठीक है, ये खुलता चला जा रहा है, पर यहाँ पे न्यू स्ट्रैंड्स बने, सीम्स लाइक जैसे एक फॉर्क के प्रॉन्ग्स होते हैं, ठीक है, जो नॉर्मल हम लोग फॉर्क यूज कर रहे होते हैं, ठीक है? सो इट हैज दिस like structure, it has these like उसमें इस तरह के अगर prongs हैं, ठीक है? So यहाँ पे they have shown you 2 prongs here, ठीक है? तो उसके जैसे यहाँ पे new prongs बनना शुरू हो गए, ठीक है? So it gives this appearance of a replication fork, ठीक है? This at this stage, this DNA molecule, it takes a shape which is referred to as replication fork, and it is just the branching prongs of the fork that contain the single stranded DNA, ठीक है? So you can see these are the branch, ठीक है? And then you can see the prongs over there. Okay, and what else you can see? This is the parental strand, so you can see 5' to 3', and this was the anti-parallel. This is 5' to 3'. ठीक है? अब यहाँ पे जो changes होंगी, तो we'll talk about what happens in these as new strands are being formed. Okay then. So let's talk about point number 3, step number 3, formation of 2 strands. How these are formed. So we talked about कि DNA helicase की वजह से helical structure break हुआ, ठीक है? Then DNA polymerase ने new complementary bases लाके attach किया, assemble हो गए. Now what happens? Now these complementary bases, ठीक है, again they have to make these phosphodiester bonds here also, because अब इनको भी तो join होना है. This was the original parental one, right? अब इनको भी आपस में join होना है, ठीक है? तो the same process of condensation will take place. So phosphodiester bond is formed in between the new nucleotides, forming the backbone of the new polynucleotide chains, because of sugar phosphate backbone तो यहाँ पे भी बननी है, ठीक है? So they've actually done this color coding है, orange color से इसको show किया, ठीक है? So these green ones are the parental strands here, right? Now this step, अब यहाँ जो आपके जो phosphodiester bonds बन रहे हैं, ठीक है? This is done by DNA ligase enzyme. So sugar phosphate backbone जो बन रही है, it is made by DNA ligase enzyme. Each new molecule is then composed of one new polynucleotide chain and one chain from its mother molecule. Okay, so this whole is the semi-conservative approach of DNA replication. Right, I hope you will remember the 3 enzymes involved. DNA helicase, then we talked about DNA polymerase, which is actually helping in making the hydrogen bonds between the complementary base pairs. और finally comes the DNA ligase, ठीक है, जो यहाँ पे sugar phosphate backbone को बनवा रहा है. Okay then. Now let's talk about this important phenomenon, leading and lagging strands. Because let's just discuss these and then we'll look at the diagram. Achha, remember we talked about the direction of formation of these strands will always be 5' to 3', right? Now, what happens? This DNA polymerase, remember these complementary base pairs are being attached there. So DNA polymerase can only build the new strand in this direction, 5 to 3. Okay, that's a rule. And DNA polymerase ही है जो उनके बीच में bonds बना रहा है. ठीक है. अब what happens as the DNA is unzipped, ठीक है, from the 3' towards the 5' end, ठीक है, because remember अगर जो आपका DNA जो helical structure था, ठीक है, जब वो like 2 उसके अंदर convert हुआ है, ठीक है, so what is happening अगर for example एक strand उसका 5' to 3', for example ये वाला strand था, so this one would be 5' to 3'. ठीक है, so what happens as it unzips, DNA polymerase will attach to the 3' position, ठीक है, of the original strand. ठीक है, for example अगर ये original strand का 3' है, तो DNA polymerase will attach here. ठीक है, it will attach here and it will move towards the replication fork. ठीक है, so you can see यहाँ पे अब इसकी movement क्योंकि यहाँ से open हुआ है, ठीक है, so it will start moving towards here because remember अब जो new strand बनेगा and we said it always forms in 5' to 3', तब जो new strand बनेगा उसकी direction ये होगी 5 to 3. ठीक है, so it will form in this direction. ठीक है, it will form in the same direction जैसे आपका ये helical structure open up होता जा रहा है, ठीक है, तो it will form in the direction of the replication fork. So इस तरह when it goes in this direction, तो this is a continuous process क्योंकि the formation it is always from 5 to 3, तो ये continuous process होगा. ठीक है, तो it binds to this, ठीक है, parental strand के 3' पे attach होता है, but then जब उसकी formation वो शुरू करते हैं complementary base pairs की, तो it is from 5' to 3'. This is what we call as the leading strand. ठीक है, so DNA polymerase enzyme can synthesize the leading strand continuously, right? So replication fork क्या था? It is the point at which the DNA molecule is splitting into 2 template strands. So यहाँ से वो split हुए जा रहा है. ठीक है, it will keep on splitting. यहाँ पे DNA helicase was acting. ठीक है, so it will bind to the 3' and then जब complementary bases के साथ hydrogen bonding करवा रहे हैं, ठीक है, so इस तरह अब जो new strands बनने शुरू होंगे तो 5' to 3'. This will be known as it will go in the same direction. जैसे जैसे यहाँ helical structure आपको open होता जा रहा है, it will start moving in the same direction. So that's why it is known as a leading strand. And then this actually is formed continuously, right? अच्छा, अब जो दूसरा strand है, what is happening in this case? Remember, DNA polymerase has to bind on the 3' area. अब यहाँ पे तो है 5'. ठीक है? So what happens? Now other template, ठीक है, it can like created during the DNA replication, we call it as a lagging strand. अब जो new बनेगा, जो यहाँ पे बनेगा, ठीक है, we will call it as a lagging strand. Now, on this strand, what happens? DNA polymerase moves away from the replication fork. Remember, it has to attach to the 3'. ठीक है, it has to attach to the 3'. ठीक है, और उसके बाद 3' पे attach होगा, फिर वो 5 to 3 करेगा. तो what will happen here? So it actually binds somewhere here in the middle. ठीक है, वो छोटे-छोटे chunks में पे collect होता है, वो continuously grow नहीं कर सकता. ठीक है, so DNA polymerase moves away from the replication fork. It means it moves from the 5' end to the 3 end. ठीक है, and it works in the opposite direction. यहाँ पे अगर it was leading strandmate was going in this direction towards the replication fork, ठीक है, यहाँ पे it has to go in the opposite direction, right? This means that DNA polymerase enzyme can only synthesize the lagging DNA strand in short segments. वो continuous नहीं होगा फिर, ठीक है, and these short segments we call them Okazaki fragments. अभी आगे जाके we'll talk about that in detail, right? But it is not a continuous process. So what happens is that it happens somewhere in the middle. ठीक है, so ठीक है, it will actually form in chunks. So ठीक है, तो वहाँ पे वो because the growth is there in 5 to 3 direction. ठीक है, तो एक short strand यहाँ पर आके attach होगा. ठीक है, और first it will let this grow. तो यहाँ से like अगर ये 5' है तो there has to be a 3' there. ठीक है, so growth होगी 5' to 3'. ठीक है, यहाँ बनने के बाद but as you are seeing कि ये जो replication fork है यहाँ से DNA helicase जो open करता जा रहा है तो ये strand खुलता चला जाएगा. तो once this will be done, और let me erase this ताकि मैं आपको एक better यहाँ से clear दूसरे color में दिखा देती हूँ. Let me use a different color here. let's use this. See, this is 3 prime. ठीक है. Now remember जो formation होगी it is from 5 prime to 3 prime. So अगर ये बन जाएगा, ठीक है, but what will happen? See, DNA helicase तो उसको बाद में खोलता जा रहा है ना, ये ओपन होता जा रहा है, तो ये it will keep on opening, तो यहाँ पे स्पेस बन जाएगा, ठीक है. तो what will happen? DNA polymerase जब यहाँ पे बाइंड नहीं कराएगा, it will have to jump and go back, ठीक है. अब जब यहाँ पे जो स्पेस खुल गया उसको भी यहाँ पे उसको भी 5 to 3 बनाना पड़ेगा. Let me use a different color for that so that एक clear हो जाए. और let me in fact erase this whole diagram and let me draw a new sketch here. See, now let's see, अब हमारा हेलिकल स्ट्रक्चर का इस तरह करके और ओपन होता जा रहा है, ठीक है, ठीक है. एंड वी सेड कि दिस वाज द लीडिंग स्ट्रैंड, राइट. इट वाज गोइंग इन दिस डायरेक्शन for 5 prime to 3 prime, ठीक है. And here in this case, ठीक है, it has to come from here 5 prime to 3 prime, right. अब what happens जब ये वाला यहाँ तक बन गया है, ठीक है, और यहाँ पे तो रेप्लिकेशन फॉर्क जो ये ओपन और यहाँ पे डीएनए हेलिकेज से खोले जा रहे हैं, तो ये स्पेस अब वेकेंट हो गई है, ठीक है. सो व्हाट हैपेंस, डीएनए पॉलीमरेज देन एक्चुअली हैज टू जंप बैक, और अब वो वापस जाके अब ये जो जगह ओपन हुई यहाँ पे फिर दोबारा उसको 5 to 3 बनाना पड़ेगा, ठीक है. तो ये दिस हैपेंस इन शॉर्ट सेगमेंट्स 5 prime to 3 prime, ठीक है. देन अगेन ये और खुल जाएगा, फिर इट विल हैव टू जंप बैक एंड देन मेक फ्रॉम 5 to 3, ठीक है. सो इट हैपेंस इन शॉर्ट क्या कहना चाहिए, शॉर्ट फ्रेगमेंट्स में होता है. So these short fragments are known as Okazaki fragments, ठीक है. So जो lagging strand है वहाँ पे continuous growth नहीं होती है 5 to 3, continuous formation नहीं होती है, ठीक है. But leading strand में there is this continuous formation. I hope it is making sense. We look at the diagram also, so it will. I hope it will become more clear then, ठीक है. So what is happening in this case? ठीक है. Now a DNA ligase will come, but remember because these complementary bases again, क्योंकि यहाँ पे भी तो sugar phosphate backbone बन रही है, ठीक है. They also have to join together. So this sugar phosphate backbone, remember the enzyme that was there, ठीक है. So this is DNA ligase, ठीक है. Second enzyme known as DNA ligase will be needed to join these lagging strand segments together to form a continuous complementary strand. अब because क्योंकि हर एक के बीच में थोड़ा ये gap है, ठीक है. So DNA ligase will come and join these together, ठीक है, because ये small segments के अंदर बन रहा है. So DNA ligase will do it then. So DNA ligase does this by catalyzing the formation of phosphodiester bonds between the segments to create a continuous sugar phosphate backbone, right. So अब इनको जो आपस में जिनके बीच में gap है, ठीक है, they will be joined by DNA ligase. I hope it is making sense, the leading and the lagging strands. Now let's talk about these, okay. Maybe it will become more clear now. So leading strand, remember they have shown you 2 strands there, okay, 5 prime to 3 prime, ठीक है, और यहाँ दिखा है 5 prime to 3 prime, ठीक है. This is the replication fork. And you can see if I draw an arrow like this, DNA helicase इसको, ठीक है, जो actual DNA है उसको open करता जा रहा है, unwind, unzip कर रहा है. How it is doing it? It is breaking down hydrogen bond between the complementary bases, ठीक है. अब what is happening? Remember, if there is a 3 prime and there is a 5 prime, ठीक है, DNA polymerase binds to the 3 prime, ठीक है. But then जब formation होगी complementary bases की, तो this strand bit, this will be 5 prime because formation is from 5 prime to 3 prime, ठीक है. So this will be a continuous pattern, ठीक है. It will grow continuously, ठीक है, क्योंकि continuously क्यों grow कर रहा है, क्योंकि यहाँ से DNA helicase इसको open किए जा रहे हैं, ठीक है. So it will keep on moving in this direction, right. So this is known as a leading strand, right. So ये continuously वो करेगा, there is no interruption in that. Lagging strand में क्या हो रहा है बेटे, this is the parental strand, ठीक है. And what will happen again, DNA polymerase जो है it has to bind to 3 prime, ठीक है. So what is happening there? So it has to repeatedly stop. For example, अगर अब ये वाला portion, let me use laser pointer now, ठीक है. If this portion अगर यहाँ पे DNA polymerase, ठीक है, bind somewhere in the middle, ठीक है, 3 like for example this will be the 3 prime, ठीक है, और उसकी जो direction होगी growth की बेटा, like it's always from 5 prime to 3 prime, right. So this is 3 prime, so 5 to 3 वो grow करेगा, ठीक है. But at the same time what is happening, DNA helicase इस वाले हेलिक्स को खोले जा रहे हैं, ठीक है. There will be space there. So ये वाला जो पूरा strand complete हो जाएगा, ठीक है. DNA polymerase then will have to come back here, जो open area है, ठीक है, and it will do the same thing here, ठीक है, 5 prime to 3 prime, and again it will keep on opening, it will do the same. So this is what we call as lagging strand. और ये जो छोटे छोटे segments के अंदर इसकी growth formation हो रही है, we call them as Okazaki fragments, right. And then later on the new strand banega, okay, it will be like catalyzed by DNA ligase. DNA ligase will actually make a bond between them, okay? It will actually join them through phosphodiester bonds. I hope it makes sense now, leading and lagging strand. So this is another example that you can see. So this is the parental strand. So you can see the 5' to the 3', and then you are looking at the direction here, right? So in this case, the direction will be from 5' to 3'. So this will be actually the lagging strand. Okay, this will be the lagging strand. And if you look at this one, okay, DNA polymerase binds to 3', and then the growth will be from 5' to 3'. Okay, so this is the leading strand. Okay, so the leading strand is synthesized in the same direction as the growing replication fork. So make sure you remember this, same direction as the growing replication fork, whereas the lagging strand is synthesized in the opposite direction. Now let's talk about these Okazaki fragments. Now these were actually like discovered by these two Japanese scientists, right? It was they actually talked about that in 1960s. So what is happening when we're talking about these short sections of DNA, we are actually talking about 150 to 200 base pairs. Okay, so this much is the length of those short sections. Okay, so it's 150 to 200 base pairs, right? These are synthesized discontinuously. So एक वक्त में इतने base pairs synthesize हो रहे हैं, ठीक है? Then, ठीक है? The DNA polymerase, it will go back. जो बाकी replication fork का area open हो गए, ठीक है? Then now they will synthesize the next base pairs then, ठीक है? So this happens discontinuously, right? So they said that these fragments are formed when DNA polymerase enzyme is working backwards, right, from the replication fork, and it has to periodically stop as it waits for the DNA helicase to break the strands apart. ठीक है, क्योंकि DNA helicase जब strands को खोल रहा है, तो उसमें भी time लग रहा है definitely. So once all of the fragments are made, DNA ligase connects them into a single continuous strand. ठीक है, or it will make those phosphodiester bonds. Right. So just to summarize, this is the whole process of DNA replication. Right, so the enzyme DNA helicase breaks the hydrogen bonds between the base pairs on the two polynucleotide DNA strands. This unwinds the helix to form two single strands, right? Each original single strand acts as a template for the new strand. Complementary base pairing means that the free floating DNA nucleotides are attracted to the base pair, and here we are talking about the activated nucleotides, right? Adenine with thymine, cytosine with guanine. Then condensation reactions occur and they join the nucleotides of the new strands together using the enzyme DNA polymerase as the catalyst, right? Hydrogen bonds form between bases on the original and the new strands, right? Then each new DNA molecule contains one strand from the original DNA molecule and one new strand, right? So this is the semi-conservative replication. And this is just as an overview diagram, just to revise. So this is a chromosome, and here they've actually shown you DNA, okay, and they have actually opened it up so you can see how it happens. So this is the helical structure, right? And this is the enzyme helicase, right? It is opening or unwinding this strand. So here we are looking at one part of the parental strand, okay, and this is the another part of the parental strand, right? So here they have shown you the leading strand. So you can see this is, okay, forming in the 5' to 3' direction. So you can look at the arrow. So it has the same direction, okay, as this replication fork, right? But lagging strand goes in the opposite direction. And you know the reason why? Because it has to move from 5' to 3', okay? So it may make all these bases, like it will keep on making these strands here. Then what will happen with it? As the helicase will keep on opening this, okay, it will have to, these don't have any strand there. So they will, DNA polymerase will go and jump back, और वो इनकी formation यहाँ से करेगा. Okay, and then these short segments will be actually joined together by DNA ligase. Okay, now let's watch an animated video about this DNA replication and about those theories also. Like we have talked about semi-conservative theory, right? So let's watch a video about these and then we'll move on to the structure of RNA.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Pyramiding starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Hi, it's Mr. Anderson, and in this podcast I'm going to talk about the Meselson-Stahl experiment. And so after Watson and Crick came up with the shape of DNA, this characteristic double helix, the next thing to unlock was how does it make a copy of itself? And so there were immediately 3 proposals as to how it might copy itself. Number 1, it could kind of unfold in the middle, and then we could copy new templates on either side. This is kind of what Watson and Crick proposed. There's another what's called a conservative theory that somehow the DNA was kind of wrapped around these histone proteins, and then it would make an exact duplicate of itself, almost like a photocopier. And then there's the dispersive model that maybe it would chunk itself into little 10, 12 nucleotide segments, and then each of those would be copied. And so what Meselson and Stahl did is they figured out which of these 3 is right. And in this picture, down here at the bottom is, it was later figured out that this is Martha Chase, who is also famous for that Hershey-Chase experiment. And so what did they do? Well, basically what they did is they grew E. coli, so this is Escherichia coli, or a bacteria, and they grew it in heavy nitrogen. What does that mean? Well, nitrogen normally has an atomic or a mass number, of 14. That means it has 7 protons, 7 neutrons. But heavy nitrogen is going to have an extra neutron, and so it's slightly heavier. And when you spin it in an ultracentrifuge, DNA that contains this heavy nitrogen is going to be moved to the bottom, and those that contain lighter nitrogen is going to stay up near the top. And so we can look at the density of the DNA to figure out what nitrogen is actually being added. And so if you think about it this way, thought experiment, let's say we started with heavy nitrogen, and then we were to copy that. So as the bacteria make copies of themselves, they're going to unzip that DNA in the middle, and since they're just in nitrogen 15, you're going to have 2 strands of DNA, but each of those are going to be all heavy nitrogen, so it'd go to the bottom of a centrifuge tube. If they were to copy themselves again, the E. coli, and make new template strands, 100% of them are going to be of the heavy nitrogen. And so what they did is they bred them in there until the point at which they were all nitrogen, all heavy nitrogen. They then switched them to E. coli living in the lighter normal nitrogen, and then they observed what happened with each generation. And so basically the lighter nitrogen in my little simulation will color it an orange color. And so bacteria on generation 0, we'll say, are going to be 100% heavy nitrogen, or 100% N15. As they unzip and make copies of themselves, that new template strand they're going to add is going to contain nitrogen 14. And so at this point, so kind of to keep track of everything, we had 100% heavy nitrogen the first time. On this first generation, now we're going to have 100% of the strands are a hybrid between the two. Half of them are heavy nitrogen and half of them are not. We're then going to go through another copying process. And so at this point, we've got one hybrid strand, another light strand, and then a hybrid strand. And so at this point, it would be 50%, if this semi-conservative theory is right, 50% light nitrogen and 50% of the hybrid. Now think in your head, if we were to do one more copy, which I don't have room for here on my simulation, well, this strand would create a hybrid, but the next 6 strands, as they split in half, are going to form light nitrogen, and this would form a hybrid. So now we'd have a 75% to 25%. And so that's what the semi-conservative would predict, and that's what they found in their experiment. So we had 100% heavy nitrogen in the first, 100% hybrid in the second, and then it split 50-50 again. This is where my simulation stopped, but then it went to 75-25. And if we were to keep going, then it would eventually drop off to all nitrogen. And so what did that tell them? It told them how DNA makes a copy of itself. It simply splits it half in the middle, and then we're going to add new template strands on either, or we're going to read that template to create new strands on either side. And so that's the Meselson-Stahl experiment. It's very elegant. It's also very important, and I hope that's helpful.
Now let's talk about the structure of RNA. So RNA, just like DNA, is also a polymer, but here its monomer is a ribose nucleotide. Remember, it had the carbon number 2 has this OH hydroxyl group attached, right? Unlike deoxyribose, which had only hydrogen, there was no oxygen there. So this has a ribose nucleotide. It is made up of a single polynucleotide chain. So it is not a double helical structure like DNA. So you will be actually looking at a single strand there, right? And here we will have adenine, guanine, cytosine, but instead of thymine, we will have uracil here, right? This is another difference. Then we just talked about 4 types of nucleotides, right? And all of these again will undergo polymerization, because remember this phosphate, the sugar phosphate backbone has to be made. As these nucleotides actually join, so it will be the same way. The phosphate group at the 5' carbon will attach to the hydroxyl group at the 3', and then they will make a 5-phosphodiester bond. And the condensation reaction will take place, water molecule will be removed. So this process of this phosphate sugar backbone formation is exactly the same, right? But RNA chain is shorter than DNA, right? So here you can look at the nitrogenous bases, and here they've shown you this adenine, uracil, guanine, cytosine, right? And remember, it's a single strand. Now here is just for you to differentiate between RNA and DNA, right? So RNA, we know, is a single strand. So you can see the nitrogenous bases, cytosine, guanine, adenine, uracil, right? And here in case of DNA, you can see there is thymine there, right? What else you're looking at is that, yes, it's a double helical structure, the DNA. And here the nitrogenous bases are exposed in RNA, right? Just a single stranded structure here, right? And it is much shorter, like than most of the DNA polynucleotides. So this is structural difference. And if you talk about the sugar would have ribose sugar, right? And in DNA, it is deoxyribose. Phosphate group remains the same in all of these, right? So that's about it, about DNA replication and RNA. And next up, we'll talk about protein synthesis.